Hi, I'm Melanie Harness, and this summer I had the opportunity to work at the Department of Population Medicine looking at uterine proteomic analysis and correlations with insulin resistance in mares. To start off with some background information for you, insulin resistance is defined as decreased glucose uptake by the cells and tissues in response to normal amounts of insulin available in the blood. This can happen by one of three ways. You can have problems with delivery of functional insulin to the cells itself, you might have problems with the insulin receptor, or there could be disruptions in the downstream signaling pathways that are important for glucose entering into the cell. So for obesity and insulin resistance, the implications that might have on breeding in mares isn't really known, but so far there have been associations made between insulin resistance and disruptions in the estrous cycle, such as having a prolonged luteal phase. For the purposes of our study, we were interested in looking at proteomic analysis between mares that have normal insulin sensitivity and the uterine proteome found in mares that have insulin resistance, as defined by proxies and reference intervals previously determined by Trieber and colleagues at Virginia Polytech Institute. We were hoping that we would see a difference between the proteins expressed in the uterus or in the endometrial environment in mares that have insulin resistance and those that have normal insulin sensitivity, with the idea being here that we can identify a specific subset of proteins in mares that are insulin resistant and possibly identify even a biomarker that might have implications on breeding in these mares. For our study, we used 36 mares in total that had either been presented to the Ontario Veterinary College Health Sciences Centre for breeding problems or mares that were part of the University of Guelph research herd between the times of May 2012 and June 2015. We collected blood samples from all of these mares and processed them for insulin and glucose measurements, which was done at the Animal Health Laboratory here at the University of Guelph. And we calculated the insulin sensitivity using this RISC, or the reciprocal inverse square calculation of insulin, and the MERG value was used to estimate pancreatic resistance. So it's a modified insulin to glucose ratio that we were using to estimate the pancreatic compensatory mechanism. For our proteomic analysis, we performed a uterine lavage by infusing 250 milliliters of saline into the uterus and collecting the fluid directly back out immediately after by gravity into the infusion bag itself. Our samples were then centrifuged and stored in the freezer at minus 20 degrees Celsius until all of our samples had been collected. Once we had all of our uterine lavage samples, we then thawed the fluid, and it was concentrated down to 0.5 milligrams of protein per milliliter for the proteomic analysis, which is then performed at the Sick Kids Hospital in Toronto by liquid chromatography mass spectrometry. In our study, we found that 30 out of our 36 mares were estimated to have decreased insulin sensitivity as according to their RISC values. 26 of the 36 mares had RISC values in the lowest quintile, or below 0.295 milliunits per liter, while 4 mares actually had RISC values below the lower limit altogether, which was 0.159 milliunits per liter. Of these 30 mares that were determined to be insulin resistant, 13 were found to have MERG values in the highest quintile, while 16 mares fell within the 5th quintile. So what this means is that 29 of the 30 insulin-resistant mares had adequate pancreatic response to their decreased insulin sensitivity. The one mare had a negative MERG value that was calculated, so she's been excluded from our study at this point. The proteomic data that we have right now, we have results available for 6 mares that we also have blood data on. Those results are summarized in the graphs that we have here. The first graph shows the RISC values in blue and the MERG values shown in red. So as you can see, mare 1403 and mare 1408 had RISC values that were above the 0.295 milliunits per liter cutoff, indicating that those mares have normal insulin sensitivity. The remaining 4 mares, for the purposes of our study, are considered to be insulin resistant. We then compared the mean spectral counts of 14 proteins that are known to be expressed by the endometrium, and that's shown in the second graph here, with the mares being insulin resistant, shown in blue, and mares that have normal insulin sensitivity, shown in red. The major function of these proteins are summarized in Table 1, and we found that the proteins that are involved in innate immunity or have anti-inflammatory effects, for example, secretoglobulin protein 1 and lactotransferrin protein had increased levels expressed in mares that are insulin resistant. At this point, we're still waiting on our additional proteomics results, and once we get that data back, we will do a statistical analysis on our data. We'll have to normalize the proteomics results first before we can apply a parametric test to our study to determine really what the association is here between the proteomes that we're seeing in the mares that have normal insulin sensitivity versus those that are insulin resistant. But to this point, our conclusions are that using the proxies and reference intervals previously determined, we found 30 of our 36 mares to have decreased insulin sensitivity in our study, but they also had adequate pancreatic compensation. The proteins that we found to be involved in innate immunity or were previously determined to be anti-inflammatory endometrial proteins in our study appear to be increased in mares that had insulin resistance. And also interesting to note, endometrial proteins that had previously been determined to be progesterone dependent in their expression, we found to be increased in the mares that had decreased insulin sensitivity, also those mares having an increased pancreatic response, therefore having higher insulin available in the blood. This supports the previous association between insulin resistance and a prolonged luteal phase found in mares, as with that prolonged luteal phase, you'll have higher levels of progesterone available, therefore causing higher expression of those specific proteins. And for example, this would be your secretoglobulin protein 1. At this point, we definitely are waiting on further results and further statistical analysis to determine any more relationships that we may be able to find or a specific subset of proteins that might be found in these mares that could be significant in insulin-resistant mares. I'd like to thank my advisor, Dr. Elizabeth Schultz, as well as the team members of the Theriogenology Lab here at the Ontario Veterinary College, as well as the Student Summer Leadership and Research Program for their funding.